So the Kavli prizes are sometimes talked about as being awarded to the biggest, the smallest, and the most complex. Now, you guys have got the prize for the most complex, probably the most complex structure in existence. Anne, where do you start analysing the brain?
Well, a lot of people start with the outer rind of the brain, the big neocortex, thought to be sort of our thinking machine. That's not where we started, though. We started right down in the middle in the old core of the brain, which controls a lot of our habits, our little rituals we form, and it's pretty important medically.
And in the sense of what you're actually looking at when you talk about the big structures of the brain, like the outside and the cortex and areas of the brain, but that's not really what your research has focused on. You've been all looking at very tiny structures and very tiny circuits within the brain.
Yeah, but we look well, in our case, we look at some very tiny circuits and some that are quite global that extend through a lot of the brain. Of course, the brain's only this big, so, it's not astrophysics, but it's just as beautiful.
More complex than astrophysics in many ways, though.
Yeah.
In terms of the number of
I think it's really wonderful
connections.
that this prize has this motto, and we've got a big challenge with the brain. It really is complicated, but it's a funny thing. I wonder if you guys agree that some apparently simplifying kind of generalizations are beginning to come. In my field, people talk about, well, there's a system for too much movement, a system for too little movement, and they're competing. It's a sort of positive, negative sort of thing.
Why don't you explain what your work is actually about?
Oh, I'd love to. So we work on the basal ganglia, and many people know about them because of Parkinson's disease, Tourette, obsessive-compulsive disorder. All of these seem to have some core problem related to basal ganglia. And at the same time, the funny thing is that we now think the basal ganglia has something to do with how we have habits, how we learn a habit, and if we ever can break a habit, how we might break a habit, addiction, big desires, reward, reinforcement, punishment. That's all stuffed into the middle of the brain. And the funny thing about it is that middle part of the brain turns out to talk to the big executive structures in our cortex, which in the first place were thought to be the really fancy ones. So it's a kind of upside down organization.
So, but in a sense, you're making that elusive bridge in neuroscience, which is to connect how circuitry in the brain works, the cells of the brain, with actual behaviors.
Oh yes, yeah. That's the tough job, and that's where we are. That is our passion.
Now, Corey, we talk about the brain as being incredibly complex, one of the most complex things in existence. The model that you've been working on is a sort of simplified version of that. Perhaps not obvious to people who don't work in neuroscience, but you actually worked on a millimeter-long worm.
Well, when you have a complex problem, the way to solve it is always to try to break it down into simpler problems. And one way you can break down a problem, like understanding the human brain, is to try to just understand one part of the brain, and then you can understand it in a lot of detail. And another way you can simplify the problem is to say, well, I'm going to keep studying the human brain, but then I'm just going to try and only study big parts of it because I know that I'll never be able to understand the billions and trillions of individual elements that make up the human brain. So the way that we break down the problem is a little counterintuitive. We say we really want to study the whole brain. We want to study the whole brain at once. We don't want to break things down, but in order to do that, we can't study the human brain. We're going to study a much, much simpler brain. So the brain of the tiny worm, C. elegans, which I study, has just 300 nerve cells in it instead of being the size of the human brain with billions of nerve cells in it. But this is an animal. It can move around, it can make decisions, it can learn things based on its previous experience, and we can really think about the whole process from top to bottom and try to understand principles that we think might also work in larger brains. So one way of sort of making an analogy is that if you wanted to understand how an engine worked, you wouldn't start by trying to take apart a Boeing 747. You would start by trying to take apart something like a Volkswagen Beetle that had a much smaller number of parts that you could kind of watch while they were moving. And if you could understand that simpler engine, then you would understand some of the basic principles about how engines work. In the same way, we think if you can understand a simple brain, like a worm brain, we can understand principles of nervous system function.
So when you see individual or specific pathways in the nematode worm's brain, how can you extrapolate them up to human behaviors in the way that Anne's work is focused on?
So the key to translating between worms and people is that the building blocks that are used to make the worm brain, which are the genes, are the same or related to the building blocks that are used to make the human genes. So the genome projects that have characterized the entire human genome and the genome of animals have revealed to us that the way that complex systems are built is to take old pieces and put them together in new combinations, not to invent completely new pieces to build the human brain. So if the genes are like a vocabulary for biology, the words have the same meaning if they're used to write a very simple book like a children's book, and as the same words have if they're trying to write the works of Shakespeare. So we try and use this vocabulary to move back and forth. We think, what does this gene do? It's sort of like, what does this word mean? And then once we have that meaning, we can start to assemble a grammar, a larger understanding that you would get by putting these pieces together.
And Winfried, if I could turn to you, a lot of your work is being driven by you inventing new technologies that enable a higher resolution of looking at neural structures. Can you, but maybe just give us an idea of what sort of techniques that you use and you've invented?
I mean, I should probably start with saying that I consider myself less a neuroscientist than a house physicist for the neuroscientists. So the gadgeteer, I like to use the analogy of Q in the James Bond movies, which of course has a very small role, but he lasts for a long time. And one could even sort of go a little bit further and say, if you think about war, it's the weapons, not the warriors, that ultimately decide the battles. So but after this somewhat grandiose, now I've always been interested in microscopy, and I'm not exactly sure. I probably came to it more by accident than by sort of deep thought, and then that sort of led me into biophysics, and sort of my first microscopy project actually was as a student in Switzerland, where I worked on near-field microscopy, sort of making a microscope that's better than resolves more than the wavelength of light, but you have to scan a small probe over the tissue and so on. And so that's or over any sample. It was very inconvenient. And so when I got to to graduate school, I got interested in light microscopy as sort of fluorescence microscopy in particular, which one should say is sort of the workhorse and the racehorse of biological imaging, because it allows us to sort of almost any microscopy nowadays. If you want any fluorescence, any immunostaining, for example, involves looking at fluorescence. But also fluorescence is the most sensitive of all microscopies. You can now image single molecules. And the thing that I did in graduate school is what now is known as the two-photon microscope, which in some sense pushes the ability of microscopy into scattering tissue. I think that's the most important part, where it's in some ways easy to image something that's very flat, like a layer of cell or a single cell. But if you want to look inside tissue, the problem is the light that comes from above and below the plane that you actually focused on will obscure your image. And you can use the confocal microscope to solve this problem, but that loses a lot of light because it actually filters out most of the light to get to the essential information, which means your signals become very noisy and you actually damage the tissue. And so it turns out the two-photon microscope does that very well. You can basically collect all the fluorescence.
So the problem being that when you're traditional microscopy, looking at a microscope, you tend to be looking at a slide or a plate with some cells floating in it. But the brain is a 3D structure.
Right.
And if you're interested in connections between layers, then it has to be a 3D
or even
model.
if you're interested, I mean, in some ways, my life can be divided into sort of my activity imaging part and sort of the structural imaging part. And the two-photon microscope was more used, even though it can also be used to image structure, it's more used to image activity inside the tissue.
Just unpack those two things a little bit for the people
Okay.
watching. So the brain looks like one thing, but it also behaves in a particular way.
So the brain is all about information, and so you have, and it's all about sort of interlocking cells. So each cell in the brain has something like between 100 and 100,000 neighbors. And this is neighbors in a meaningful, neighbors that it actually talks to. It has so-called synaptic connections with them. And so taking the brain apart, I mean, I'm talking about a mammalian brain now. Taking the brain apart basically severs a lot of these connections, maybe all of them, and then the whole function is disrupted. So you have to basically look at the signals, the activity of the brain while it's intact. And so you need a way to look into the brain that can actually image the intact brain. So one way, in some sense, it's less of an imaging technique, but you can take an electrode, a very thin wire, stick it into the brain. But that has its own problems. And if you want to sort of look, for example, for calcium activity, where you have calcium is a messenger that's used in all cells, and it's a sort of universal messenger. And you can there are fluorescent dyes that change the fluorescent properties, so they become brighter as the calcium goes up. So you basically have a signal that is a chemical signal, but it actually is very closely related to the electrical activity of the brain. And so in order to, say, image a cell that's 500 microns down into the brain, you have a cell body sitting there, you want to know what it does. So you somehow get the dye in there, and now you want to see its activity. So so you have to focus down there. You put the focal plane down there, but then the dye rarely is only in one cell. There's dye in sort of the whole brain, and so you have to isolate that one cell. And so you can do this by putting your focus down there, because the two-photon effect works in a way that it only really occurs in any appreciable rate if the intensity is very high, because two photons have to be at the atom at the exactly same time, and exactly I mean within 10 to the minus 15 seconds, so within a femtosecond basically, and that only happens if you have huge intensities, and then you get fluorescence excited, and then the light comes out, and then you can and the amount of light that comes out tells you whether the cell is active or not, and so this allows you to look not quite as deep as and I think Anne's problem in some sense, or in some of this, is she picked a very difficult part of the brain because it is so difficult to get to. Electrodes are okay, but imaging, she's safe from us microscopists. You could say.
No. So our goal now in this coming year is to use this kind of method, calcium imaging, way down deep. So we are completely, totally fans of Winfried Denk and everything that he's done. And
it strikes me, listening to all three of you, that this is the field of research that I came from 10 years ago, but it has changed so significantly that I can, what you're talking about techniques that I don't think anyone had really even considered.
And
back then.
a lot of us, now you're different, you've been doing molecular biology for a while, but they keep changing your methods. And for our lab, we do totally different things than we used to do, and we're changing all the time. We're surfers. We're riding the wave of new techniques, because it's the only way to solve a problem. Our techniques used to be so primitive. That's what Winfried was saying.
How much do you think the field has been changed purely by technological achievements, by inventions of new techniques? Kari?
Oh, I think that technology drives science in biology, in physics, in every aspect of science. I think that's part of the astrophysics Kavli prizes as well was the use of CCD cameras to solve problems for the first time. And now everyone, this incredible technical breakthrough from 20 years ago is something that now everyone carries in their wallet and their little camera in their cell phone. So it's when you have a new tool, when you have a new instrument, you can see something for the first time. And a brilliant person can just with their mind see the same thing for the millionth time and make an incredible insight. But it's an awful lot easier to be the very first person to see something and to make a brilliant insight. And technology pushes forward everything that we do.
Okay.
Can I just say, it's so great for young people who are just starting out and deciding whether they want to be scientists, because I think what they can expect, I guess you guys might agree, is that, they're just going to be, I hope, more and more new techniques. So we're sort of, it's a straight, I feel like a very early pioneer in this whole period.
I think that's what I mean about how the field
it's pretty
having
exciting.
changed so much, because neuroscience is not, I mean, it's been around for a good century or so, but it feels like it's gone through a major sort of tectonic shift in the last decade. And I feel like a lot of that is technology driven, right? Is that your perception?
I think there's been, for example, a great change in the way that molecules and genes are used in neuroscience that started about 15 years ago, where prior to that, people would study genes to study a gene, and now people use genes as tools to probe the nervous system, to monitor activity in the nervous system, to manipulate activity in the nervous system. And that's really that's changing molecular biology from a science to a tool for neuroscience. And that's what Anne is alluding to, for example, in her work. So that's one of the large changes that started maybe 15 years ago, but has really accelerated massively in the past 10 years.
I mean, I think, one shouldn't forget that all the tools are in some sense based on somebody's scientific discovery. I mean, now, if you look at physical tools, physics-based tools, then most, say, neuroscientists don't, in some sense, know the history of those tools that are in physics, because that's not something that's part of the neuroscience canon, while, say, molecular biology is part of the canon, and so the connection becomes clearer. And that makes it, in some sense, easier for sort of mediocre physicists like myself to make an impact in neuroscience by sort of basically knowing a little bit of physics. I mean, if you think about for example, two-photon absorption, which was predicted by Maria Goeppert-Mayer in 1931 in her PhD thesis. I mean, it took another 30 years to actually do it experimentally, because you needed a laser to actually experimentally see it. But it wasn't actually, at the time when we started working on it, it was not cutting-edge physics. But it's still, it's one of those things that sometimes sort of knowing something from a very different field is very useful. But nonetheless, of course, it was when it was discovered and studied, it was scientific insight. It was not an engineering thing. Nothing starts at engineering. Everything starts at science, which is basically what Corey was saying.
And, we mustn't forget that when these new techniques are used, and people begin to get a new insight about the brain, then that new insight in turn changes everything in the field. And we've seen that. I mean, surely in the time I've been in neuroscience, it's what people say about the brain, or brain and behavior, as you brought it up, is so different now than a few years ago. And we couldn't have done it without the microelectrode, and now without these imaging methods and molecular biology.
Okay, I'm asking all of the laureates this question, and it's a bit of a fantasy one, but it's
Fancy?
Fantasy.
Fantasy.
I suppose it's quite fancy as well. So imagine, I want to ask each one of you in turn, but imagine that the resources are unlimited. You've got, you don't have to apply for a grant evermore, and you've got infinite amounts of money. What would be the one big experiment that you would dedicate all of that free resources to?
The first thing I'd do is faint if I don't have to apply for grants, which is a lot of what we have to do in science these days.
After you'd woken up.
Boy, that's a toughie. So Okay, I mean, you're really getting to it. So I'm the deepest advocate of fundamental research. It's just totally, everything rests on being able to do that. Everything, a lot about how we can get on in this world of ours. But, you know, it would be very nice to help people. It would be so very nice to help people. So I would use my magic wand, and I would go on the basis first of our field where I know things the best. I would systematically try to use this knowledge to help the people who have disorders related to the basal ganglia, all the way from neuropsychiatric problems to kind of motor problems. And that is not to put down the basic science. We're not going to probably do that without the basic science. But I think that's a pretty good goal.
Corey, you've got a blank check. Well, what do you do with it?
I think the first thing to say is that if I really had a blank check, I wouldn't want to work by myself. I would want to bring people around me and have a group of people working together on an exciting question. And if it really didn't matter that something had to happen in 5 years or in 2 years, I think I would try to see if we could understand the basis of some of the unique human abilities like language and music, and try to think about what the animal origins of those are, and to take what we know now about behavior and brain systems and study these fascinating, important, at some level not medical, but critical elements of what it is to be human.
So both small topics. You're going to cure disease. You're going to understand what it is to be a human being. Winfried, what would your dream project would be?
I mean, it's slightly embarrassing, but I don't think I would do anything different. And the reason is that I am in some ways in the situation, almost in the situation that you described. I mean, I have not unlimited funding, but I have enough funding to basically be limited by my imagination rather than by resources. Now, there's always, at some level, a resource limitation, and I would probably, if I really had sort of unlimited funding, I would probably scale up some of the things I'm doing maybe a little bit faster, sort of try to find people that could join me. I mean, it's always the most important thing is to be surrounded by people one can talk to in an intelligent way about what one is doing. But I think that's the unique thing about Max Planck. I mean, I think that's what makes it in some sense. And it does allow you to do that. And there's no 5-year thing, there's no 2-year thing. There's just, I mean, at some point you have to retire. But until then, you basically do whatever, and if you think it will take 10 years, I mean, basically, I had basically 9 years between my last 2 major papers. So, I mean,
I think you've just made a lot of scientists very, very jealous, envious of your position.
But partly, I think, with a good reason, because I think people still turn us down. I mean, turn Max Planck down, which I think they just don't understand how wonderful it is in some sense. And if you have this sort of really long-term goal. I mean, if you want to sort of run a broad group, a lot of sort of incremental things, then that's not necessary. But I think I've really felt I've been extremely lucky in my career. I've been at Bell Labs, which had similar advantages, and I've been at Max Planck now, and I have a little side appointment at Janelia, which also has some of that spirit. I mean, it's not quite, there is a little more pressure to perform, but I think it has the spirit of try something that's impossible and just don't try things that you know are working. And so I'm trying other things now that I don't know whether they're working, and they should have a sufficiently small probability of working to be interesting.
Well, I think on that note, I say thank you to you all, Winfried Denk, Corey Bargmann, and Anne Graviel. Thank you very much.
Thank you.